Okay, in our last video, we went over the various reactions involved with carbohydrate metabolism. This video will start off with the energetics of glycolysis, okay, and then we will move on from there. So, the energetics. Okay, so if we analyze, okay, the direct analysis of the changing Gibbs free energy, okay, for these various values or for these various reactions, it doesn't readily explain the pathway. The metabolic. Okay. And it definitely does not explain the efficiency of that pathway. I spell efficiency. Leave like that. Okay. As we've discussed before, we have to account for these cell conditions. Things like the pH of the cell, the water content, the ions present, and so forth. How much ATP is there in the cell? How much ADP? How much AMP? How much inorganic phosphate is around? All these sorts of things. Okay? Once this is done, we can see that reactions 1, 3, and 10 are the only ones with significant negative delta G values. Okay? And so these are also the irreversible reactions. Okay? All the other reactions, okay, and specifically 2, okay, and then 4 through 9, their change in the Gibbs free energy is on the order of 0. Okay? What does that mean? That means those reactions are close to equilibrium. Okay? And that also means that they are reversible. Okay? They're reversible without much effort. All right? You might wonder, well, is this happening constantly in my body? And the answer is no. Okay? So glycolysis is regulated by way of allosteric control of the hexakinase enzymes. Okay, so the kinetics of the hexokinases And allosteric regulation of the enzymes in reactions 1, 3, and 10. All right, pop quiz. What are those enzymes? If you don't remember, go back and look. Okay. Not only do I want you to be able to know those enzymes, but I want you to know what does it mean for those enzymes to be allosterically regulated. Explain that. Okay. Define that. Okay. So, hexokinases. Hexokinases. We have 4 of these different proteins. Okay. So we're going to go over each one. Okay. Hexokinase 1 is a ubiquitous enzyme, which means it's found at least in some concentration pretty much everywhere in your body. Okay? But highest concentrations of this particular enzyme are found in the brain and kidneys. Okay? So hexokinase 1 is most concentrated in those 2 organ systems. Okay? Hexokinase 2, all right, is principally found in insulin responsive tissues. Okay? Those would be skeletal muscle, and the adipose tissue. Okay? Skeletal muscle, I think, needs no further explanation. Adipose is a term you might not be familiar with. Adipose is the fat cells. Okay? So an adipose cell is where glucose is converted into longer chain, longer chains of things like glycogen, so that it can store energy for future use. All right. Hexokinase 3 is also ubiquitous. All right. But its overall abundance is low. Okay. It's everywhere, but there's not a lot of it. Okay. Tissues in which it is most concentrated. Okay. It's most concentrated in the following: the liver, the lung, and the kidney. Okay. So you can see that hexokinase 3 is really focused in our tissues that are being used to, help provide energy and clear waste. Okay. Hexakinase 4 is kind of an interesting one. Okay, it behaves a little differently than its three cousins here. Okay, it is mostly found in the liver. Okay, and the pancreas beta cells. All right, the intestine, and the brain. Okay, that's the 4 locations for hexakinase 4. Hexakinases 1 through 3 have very high glucose affinities. Okay, very high glucose affinities. All right, they are half saturated. Okay, at blood glucose concentrations on the order of 0.1 millimolar. Okay, 0.1 millimolar, right? Most of the time, your blood glucose is around 4 to 5 millimolar. Now, I say yours because we know that doesn't apply to me. As a diabetic, my blood glucose concentration is sometimes significantly higher than this. Okay? So for you all, your hexakinase 1 through 3s are totally saturated. Mine are beyond saturated. They're just straight up overwhelmed. Okay? They don't know what to do with themselves. All right? And these are inhibited by their product. Pop quiz, what is their product? Pause the video, name it, then see if you're right here in a second when I write out the answer. They're inhibited by their product, glucose 6-phosphate. Okay? Hexakinase 4 is different. Okay? Hexakinase 4, it requires a much higher blood glucose level. Okay? Something around the order of 10 millimolar. Okay? And it's not inhibited by glucose 6-phosphate. Okay? The primary function of hexakinase 4 is to divert glucose storage. Okay? Into glycogen. All right? So hexakinase 4 says, oh, there's a lot of glucose around. I'm going to start getting to work. Okay? And it doesn't stop because G6P means nothing to it. All right? Unlike the other hexakinases where glucose 6-phosphate, they see enough of it and they're like, yeah, we're good. We're good. Cell's got enough energy. We're all right. Okay? Here, not so much. Okay? hexakinase 4 says, haha, we need glycogen. Let's make glycogen. Okay? And it is regulated, okay? It is regulated by I believe it's glycogen kinase response protein. Okay. All right. And that protein is sensitive to fructose 6-phosphate levels. Okay. So in situations where the cell has enough energy, it's got plenty of glucose 6-phosphate, the hexakinases 1 through 3 have shut down. Hexakinase 4 is still churning away, right? But the cell is going to have a lot of fructose 6-phosphate sitting around because it's going to convert glucose 6-phosphate to fructose 6-phosphate. That's going to turn this protein on, which is going, off. It's going to inhibit its ability to bind to hexakinase 4. Okay? And so hexakinase 4 will take all the excess glucose that is beyond the need of the cell and convert it to glycogen for energy storage. Okay? If fructose 6-phosphate is low, then these hexakinases might be shut down. This one will not. But this glucokinase response protein will be active. And so it will stick onto hexakinase 4, thus preventing the glucose that is still needed to produce energy for the cell from being diverted into glycogen storage. Okay? All right. And of course, this isn't the only control mechanism. Okay? We also have allosteric regulation mechanisms. Okay? And these allosterics function for the first 3 hexakinases. Okay? PFK1 and PK. All right? For hexakinases 1 through 3, we've already discussed, their allosteric regulator is glucose 6-phosphate, their product. Okay? For PK, and this also includes PFK2. Okay? For PK, okay, high concentrations of AMP will function as the inhibitor. Okay, will function as an activator. Okay, so in other words, you can think of activators as kind of like negative inhibitors, where inhibitors will slow the function of an enzyme. Activators will enhance the function of an enzyme. It'll kick it into high gear. Okay. ATP, acetyl-CoA, alanine, and long-chain fatty acids. Are inhibitors. And this makes sense, right? Because if there's a lot of AMP sitting around, that means the cell needs energy. And so PK, phosphokinase, is going to start kicking into gear. Okay? If there's a lot of ATP, a lot of acetyl-CoA, a lot of alanine, a lot of long-chain fatty acids, these things all imply okay? Or sorry, no, not alanine, alpha lauronic acid. My goodness, okay? Alpha lauronic acid. Alpha lauronic acid, and other long chain fatty acids. Okay, these will inhibit it because these molecules indicate that the cell has plenty of energy. So phosphokinase doesn't need to function. Okay, PFK1, all right, is the most carefully regulated of the enzymes we discussed. Okay, so the most carefully regulated. Its inhibitors include ATP, PEP, and citrate. Okay, PEP is the end product, right, of our glycolysis, and citrate is one of the products in the citric acid cycle. ATP is a process of all of these. So imagine that. It's inhibited by the products that are produced that say, hey, I've got plenty of energy to the rest of the cell components. Okay? But it is activated by AMP, which I'm sure does not come as a surprise because if there's a lot of AMP in the cell, that means that the cell is starving. Okay? PFK2, okay, is a bit of a strange one. Okay? Its function is controlled by the presence or absence of inorganic phosphate. Okay? So, when it is phosphorylated, okay, PFK2 functions as a phosphatase. Okay? When it is not, okay, it functions as a kinase. All right? This function here is controlled by the hormone glucagon. This function is controlled by the hormone insulin. Okay? And that functions as an excellent segue to our other method of metabolic control, which is hormonal. Okay? So we have hormonal control or regulation in our cells. Okay? There's primarily two hormones responsible for this control. All right? The first is glucagon. Okay? Glucagon is released by pancreatic alpha cells. Okay? When the blood glucose level is low. Okay? This activates PFK2 and it decreases the activity of PFK1. Okay. It completely inactivates liver-found phosphokinase. Okay. So glucagon, when it's released in response to a low blood glucose event, activates PFK2s, and it specifically activates the phosphatase function, right? Shuts down PFK1 and shuts down PK. All right. And so that allows us to, our body to rapidly switch into a metabolic state of breaking down glycogen and other energy stores. Okay. Insulin is the sister hormone for glucagon. Okay. So insulin is released by pancreatic beta cells. Okay. In individuals with type 1 diabetes, typically what is found is that the beta cells either do not produce insulin at all, or the insulin they produce is mutated in some way to make it ineffective. In individuals with type 2 diabetes, the beta cells produce insulin. That insulin is normal, it functions the way it's supposed to, but it either they produce insulin at a lower rate than what they should, or their functionality has been burned out by fat invasion, is the most current theory. Because as you gain weight, especially around the middle, your pancreas can become invaded by adipose tissue. And that adipose tissue can crush or otherwise invade the space for beta cells, thus preventing the release of insulin. And so as a result, your blood glucose levels stay high. Okay. All right. These activate, or insulin activates, the kinase function of PFK2. Okay. And the muscle and adipose tissues they have possess insulin sensitive receptors on their cell membranes. Okay. And that enhances glucose transport. Okay. So whenever your blood glucose level is high, say after a meal or after some kind of sugary snack or sugary beverage, right? Your pancreas beta cells release insulin. That insulin then kicks over to change the function of PFK2 from a phosphorylase to a kinase. And then PFK2 starts to send things down to be stored as glycogen. The muscle and the adipose tissues, they recognize the insulin in the bloodstream, and that opens up their glucose transport mechanisms. So they start to take on as much glucose as they can handle. Okay, which is why if you've ever experienced a low blood glucose event, right, which is something that diabetics can deal with, especially diabetics on uh, injected insulin. Okay. They can have low blood glucose events, and there is a very significant feeling of weakness. Um, but imagine right after a heavy or a strenuous workout for you, since the four of you are very healthy individuals, right? Um, right after that strenuous workout, your muscles feel a bit weak. They feel tired, right? You can feel the burn, so to speak, right? Um, and everything feels like it just takes inordinate amounts of energy. Everything seemed heavier, right? That is what a low blood glucose event feels like, right? And the way that you address this is you typically have something that has some easily digestible carbs in it, okay? And you can feel shortly after that, you feel the energy flooding through your body. You can feel your muscles taking in the glucose. Um, it's a very near electric feeling, and if you've been without food for a couple of days, you can feel this too. It's called being food drunk. It actually makes you a bit giddy, um, as though you had been intoxicated. Um, it's a very odd experience. It's a very odd experience. Um, I would not recommend it. Don't go trying it. All right, don't be crazy. Okay, please. But now you know. Like, there really is a thing. There really is a sensation called being food drunk, and that is related to going from an extreme low blood glucose to a higher blood glucose in a very short period of time. That will finish up our recording for today, or our video for today. We'll very briefly discuss some glucose-induced gene expression before we move into glycolysis. And I will then move into nucleic acids after that. Hopefully, we will be able to get through nucleic acids before the end of the semester. They're very important biomolecules. They happen to be my favorite class. That may or may not appear as a bonus question on your next exam. So, just throwing that out there. All right. Good luck. I look forward to reading your NMR reports.